आइए अब हम बात करते हैं रिएजेंट्स की और सप्लाईज की जिनकी हमें ज़रूरत पेश आएगी फॉर प्रोटीन प्योरीफिकेशन फ्रॉम इंक्लूजन बॉडीज़। जैसे कि एक स्टेप्स के हवाले से हमने बात की थी, व्हाट स्टेप्स आर गोइंग टू बी फॉलोड टू आइसोलेट प्रोटीन्स फ्रॉम बैक्टीरिया। अगर उनको लेके चलें तो हमें एक आइडिया मिल सकता है व्हाट काइंड ऑफ बफर्स वी नीड। Variety of buffers are required. We will need two types of cell lysis buffers. Then we need some detergents like deoxycholic acid. This is a bile acid. Then we need an acid HCl, strong 12 molar acid, which will be used to adjust the pH to bring the pH from more basic towards acidic. Then inclusion body solubilization buffer 1 and 2 if at the end we need to solubilize the inclusion bodies. Composition के हवाले से cell lysis buffer 1 or 2, they are more or less similar. The only difference is the concentration of EDTA and presence of tritone in cell lysis buffer 2. Rest of the composition is same, tris HCl, EDTA and sodium chloride, and these buffers are prepared at pH 8 and they can be used further for cell lysis. Inclusion body solubilization buffer, tris HCl, EDTA, sodium chloride, urea, PMSF. सेपरेशन के लिए या आइसोलेशन के लिए जो इंक्लूजन बॉडीज़ की है उसके लिए हमें आगे चल के दो मेथड्स अवेलेबल होंगे और उन मेथड्स को सामने रखते हुए हम अपने बफर्स को डिसाइड करेंगे विच बफर्स वी आर गोइंग टू यूज़। एक मेथड में ट्रिटोन X100 यूज़ होता है, दूसरे मेथड में यूरिया यूज़ होती है। सो इन दिस सिचुएशन वी आर नॉट गोइंग टू यूज़ ऑल ऑफ दीज बफर्स, वी विल डिसाइड later on which protocol we are going to follow and which buffers are required. Solubilization buffer 2 contains potassium hydrophosphate, KH2PO4, EDTA and sodium chloride. Then KOH, potassium hydroxide, is required. This is required to maintain the pH towards the basic level. During the incubation period at different steps, sometimes the pH changes of the extract or of the lysate, and that pH needs to be maintained. For that purpose we need to have this alkali KOH, and we need PMSF and 1x 2x SDS gel loading buffer. जेल लोडिंग बफर की ज़रूरत पेश आएगी हमने जब एक्सट्रैक्शन कर लेनी है, देन वी हैव टू कंपेयर डिफरेंट डाइल्यूशंस that which dilution have our required protein of interest. For that purpose we may have to run a gel SDS page. For that purpose we need these buffers. Then we need tris Cl with urea. In this tris Cl with urea is required if we are following a method that involves urea. In this case we prepare different concentrations of urea, and these tris urea is going to be prepared very freshly, otherwise urea is going to be metabolized and previously prepared or older solutions, they will not give the best results. Then we need some enzymes. DNase 1 is required, so that if there is sufficient amount of DNA present in the lysate we can degrade it. Lysozyme is required, which may be used as an option to lyse the cells, to mechanically or to enzymatically disrupt the cells, and these solutions are prepared in tris Cl. SDS page will be required 10% to test the extracted protein samples. Centrifuges and rotors are required as per the sizes of the tubes. Microfuge can be required, or some other different centrifuges with different routers are required. Special equipment में देखा जाए तो pH paper आएगा, because we have to monitor the pH during all the process so that we can maintain the pH. So pH paper and gloss rod is required. Vector or bacterial strains which are basically the source of proteins. vectors that are recombined having foreign gene inserted into the bacterial cell. That bacterial cell has expressed it. Now the time is to extract the proteins, and those E. coli cultures are going to be required so that we can have this sample, bacterial sample, for extraction of the proteins or for the extraction of inclusion bodies from where we can isolate proteins.